Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome back to the Genetics Podcast, everyone. I'm very excited to be here today with Dr. Euan Ashley, who's the chair of the Department of Medicine at Stanford and co-founder of a number of companies and really one of the pioneers in the field of medical genomics. He's involved in some of the earliest tools for interpretation of the human genome, has set a couple of records that we'll talk about in some areas of medical genomics. And he also wrote a book, The Genome Odyssey, Medical Mysteries and the Incredible Quest to Solve Them. I've always admired your work in the way that it cuts across disciplines as well. So we're going to talk a little bit about, the breadth and different pillars of what you do, because I'm a firm believer that the most interesting science sometimes happens at these intersections where you get people together from very different worldviews, and that's where the magic happens. So thank you for taking the time to be with us today.
Yeah, thanks, Patrick, for having me. Big fan and excited for the conversation.
I want to start with some of the interesting cases or research from your career that you feel like have really shaped you. You've worked across a number of areas, and as I mentioned before, you've done quite a few interesting things. But I'd love to hear one or two of the stories that maybe encapsulate the way you think or some of the impactful moments you've had along the way.
Yeah, I mean, I think one of the joys and privileges that we have as people who try to do science and medicine and combine the two and have the two inform the other is that we do have these pivotal moments, I think, where something has happened in one of those lives, the science life or the medical life, that really changes the direction or opens the door and sort of points in a particular way. Certainly, we've spent a lot of time over the last decade and more thinking, of course, with the application of whole genome sequencing to medicine and working on the aspects that we saw as really necessary for the move from a technology that has been so important for discovery and understanding of just the human genome and its importance for disease, to the point where we could use it as a clinical medical test. And I think it wasn't obvious at the beginning what those things were. So those have been the areas that we focused on. So many of the stories I think of that have become pivotal to our journey really relate to elements of either quality of genome sequencing or speed. Of course, we all show the cost curve, and that's been incredible too, and the ability to get base pairs for just less and less money has been really important in what's driven the field. But I think for the work that we've done, it's really been stories that speak to the power of genome sequencing for changing the trajectory of a patient and the importance then of quality or speed. And I'm sure we'll talk a bit more about speed, but thinking about some of the earliest opportunities, I mean, there was a time when it really wasn't. It really was a months to years operation to think about both sequencing a human genome and then interpreting it. And certainly the first one we did in 2009 really was, 6 to 9 months of like 25 people doing nothing but really focusing on it. But it wasn't that much longer after that we had a group come to us with a challenge, which was that a small baby who'd been admitted to the neonatal intensive care unit at Stanford, was delivered a little early with a low heart rate, had essentially been having multiple cardiac arrests, and each time the staff would have to come and do CPR to bring the patient back. It was obvious pretty early that the little baby had a version of long QT syndrome, an inherited condition affecting the repolarization of the heart, and so causing this long QT interval, QTs on the electrocardiogram, but predisposing to these cardiac arrest rhythms, and saying, is there anything you can do? We think we know pretty much this is a genetic disease, but we don't know what the molecular basis is. And we really would change our therapy if you could tell us, is this a potassium channel form of the disease or a sodium channel form of the disease? And, there'd been a handful of genomes interpreted for medical reasons at that time. Very few of them had been done all that quickly, although Stephen Kingsmore, who's really been a pioneer in this area, had shown that it would be possible in about 50 or 60 hours to do a form of genome sequencing plus a form of interpretation. So we called up the team at Illumina actually and asked if they might be able to help. And they said yes. One of the incredible things about being in this world is just how willing people have been to lay down whatever else they're doing and focus on the one thing. And I guess there are a few things in life more tearing, more capable of tearing at your heartstrings than a little baby suffering from cardiac arrest. So yeah, folks in Illumina, folks in one of our startup companies, Personalis, contributed. We talked at one point to Gilead, pharmaceutical company, and it was all hands on deck. And then I remember that one weekend after we'd sent the blood sample in the Illumina lab and basically said, sure, we'll put a whole machine on this over the weekend, that he basically, the postdoc in my lab, James Priest, actually pretty much rewrote the software to try and speed it up even more so that the moment the data arrived, which was just after the weekend, we could interpret it as fast as possible. And in fact, pretty quickly, we ended up finding it was a potassium channel and being able to, a potassium channel version of the disease and being able to give that back to the bedside team. So just one example, and that little girl's now, I think, 7 years old, and she had a defibrillator implanted. She was one of the youngest people ever to have that done to help save her. But being able to identify the molecular basis of her disease and then for having a precision medicine applied at the bedside for someone who was days old was just a really pivotal moment for me in terms of the power of genome sequencing for the information it can provide for patients with genetic disease.
Yeah, that is amazing. Where's the ultra rapid whole genome sequencing in critically ill infants gone since then? That is one of the Guinness Book of World Records I alluded to, because you did that, but then you all thought about how do you actually scale this and make it part of more of a accessible clinical genomics workflow, right?
Yeah, exactly. I mean, I think that was, in a way, the itch at the beginning. We realized the power of that, and that was with Illumina, with the Illumina Clinical Lab at the time. And Stephen Kingsmore, of course, continued to do great work in this area, including demonstrating clear cost effectiveness of doing this kind of work. But later, we felt that, that we'd be able, that's an opportunity to go faster. And we'd been working with the Nanopore platform, and we're impressed at how much it had advanced in terms of quality. And then the idea that perhaps actually there was a route here to very rapid sequencing. So obviously for Illumina, you're mostly talking about 18 to 24 hours minimum, and then a bit more obviously for interpretation. With Nanopore, especially, they had a machine called Prometheon, which is just a great title for a sequencing machine, 48 flow cells in one. And I mean, this thing can do in basically a minute a 1x genome if you put the same DNA on all of the flow cells at once. Of course, we're looking for a 60x plus genome, so it took about an hour, but that's pretty fast. And then the issue was that the machine next to it that came, not surprisingly, with just a small number of GPUs in it, wasn't really capable of dealing with that data from all of those nanopores at one time. And so an electrical engineer, Sneha, who worked in this, Goenka, she had basically built this kind of real-time pipeline where basically the data would be pushed to the cloud. And one of the key things, I mean, it's not kind of a rocket science insight, but nobody had done it to that point. And she really came up with the plan where each of the algorithms that we use, and your listeners will be quite familiar with the variety of algorithms that are used for interpreting human genome data. I mean, some are optimized for CPUs, like a general central processing unit in a computer, but some run in parallel, are like more deep learning or AI type tools that are optimized for GPUs, graphical processing units. And so she was making sure then that the data was being parsed into put into parcels, sent to the cloud, and then the compute that was being applied was optimized for the algorithm that it was being done. So basically, the overhang after that 1 hour of sequencing went from 18 hours, which obviously is too much, to about 30 minutes, which was just a really huge change. And so then it became, how fast can we go from getting the blood sample through to making a diagnosis? And there's other parts of that. And, Stephen had shown that down to, I think, about 18 hours on an abbreviated Illumina platform. But also, that would be hard to do every single time. We were interested in something that could be scalable. And we showed over the course of that study, and not to go into too much detail, but that we could essentially do that regularly in under 8 hours from blood sample to diagnosis.
Wow.
And so that, I think, from our perspective of thinking about ourselves, and I'm also a physician working in ICUs for adult care, not NICUs, but that is a game-changing sort of number, because now, within the course of one nursing shift, you could send a blood sample and get back a whole genome sequencing result. And that I think for these little babies in particular really ICU anywhere, but the little babies in particular could be potentially game changing.
What does it take for that to go from where it is now to be in every neonatal ICU where because you have the nanopore, you have the IT stack and informatics you're talking about. What else needs to happen?
Yeah, it's a great question because one of the things that we really want to do is to get this to every baby who would need it, and I think that definitely, I think Nanopore are poised and happy, of course, to place machines wherever anyone would want them. In the aftermath of that in the year or two since we did that work, we've worked with a bunch of people around the world actually who've called us and said, how could we do this? And the original software is open source. And as you said, as long as you can get the machine next to the patient and the workflow is actually pretty straightforward, the data can then be piped to the cloud and everything else can then happen in the cloud. So then I think it becomes a question of what is the business around that, because as an academic lab at Stanford, we can't service the entire world. Obviously, we can service Stanford, but then I think we and others are looking at what does a scalable business around that look like, and how can we make it as accessible as possible? And then you get into the challenges, which we could go into, but are probably less interesting to your listeners around, what is a diagnostic test, a genetic diagnostic test? How is it reimbursed for inpatients versus outpatients? And there's a lot to say there, but it's a bit maybe of less technical interest around genetics, but it's really important to the scalability and the answering of your question.
Yeah, we could circle back to that if we have time, but based on everything that you do, I suspect that we won't. But there's a related question to that, which is around not critically ill infants, but there's a whole wave happening around newborn screening, across healthy babies or presumed healthy babies until the genetics tells us otherwise. I'd love to get your perspective on that, because that, going back to my original interest in genetics, I've always been fascinated by the idea of what if we had this from day one and could think about this as an informatics problem over our lifetimes versus you get testing when you need it, but you miss all the other stuff. I'm really curious to get your perspective on that.
Yeah, no, it's a great question. Obviously newborn screening since Robert Guthrie and the little piece of cardboard with the heel prick blood spots on it has been pretty much accepted. I think at the time it actually was fairly controversial, but it became readily accepted. Of course, it's been part of everything we do. It's obviously a surprisingly very old technology that is used to detect, essentially, in the most cases, genetic disease. And I think it's been really interesting when Wendy Chung's study is probably the most recent and largest one from New York, demonstrating the utility of whole genome sequencing as a much more modern and I think, clearly much better approach and clearly much more modern approach. The things that we've been wary about since the beginning of genome sequencing are the same things that people are wary about in replacing traditional biochemical-based screening with whole genome sequencing for newborn screening. And that is, you get a lot of data, and it's relevant not just to the things we check for in current newborn screening, which varies in the US, at least across states and across countries in Europe and the rest of the world. But here you clearly get the whole thing. You've got the 6 billion data points that speak not just to those more limited diseases that are a bit more biochemically genetic based, but all the others, including things like your disease risk for Alzheimer's disease and things that would happen to you, much degenerative diseases that have a genetic component that would happen later in life. But I think that we have a framework and a structure and a paradigm for dealing with that in a way that wasn't true 10 or 15 years ago. I think very many colleagues around the world have thought carefully about the ethics of this, and it's just become much more available. So people are much more comfortable with it. And we're just better at understanding variants of known and unknown significance. We have much larger population genetic studies that help us inform that. We now have variant effect maps, cellular maps for every single variant across several genes. And increasingly, we'll have that for almost all the medically relevant genes. So I think the foundations are really well built now for that transition. And then your point goes further, which is that what is the utility then, not just to replace newborn screening for those things that are currently relevant for newborns, and obviously PKU being the initial trigger factor for that, and things that could be devastating if not caught at a very young age and where there's a clear intervention, but things that are relevant across the entire lifespan. And I'm a cardiologist. I see adult patients with heart disease, and particularly we focus, of course, on genetic disease and inherited cardiomyopathies and others. But just thinking of the largest number of people in the world who they die of cardiovascular disease. And we understand quite well now that the site frequency spectrum of the risk for cardiovascular disease, including polygenic risk scores in the long tail, Mendelian disease in the larger, with the high effect, large effect variants, and that entire spectrum in between. And I think that when we think about that disease, that we think of a disease of middle age, or a disease where you don't get recommended to have your cholesterol checked until you're maybe 40 years old.
Yeah.
But your genetic risk for heart disease begins quite a lot earlier, 40 years, in fact, almost 41 years before that moment. And so maybe you could argue we don't want to be burdening children with that knowledge, and they don't need to be burdened with the knowledge if they don't want it. But there does come a moment in life, probably before you have your first heart attack, when it would really make sense to know what both your genetic and environmental risk for that disease would be in case you want to intervene. And so I do think that we're just in a better position to understand the sort of ethical basis of it, and really having our arms around how to deal with the data in a way that people can decide what they want and when they want it. And I think that's different and has changed and puts us in a good position for more preventive care.
Yeah, I agree with that. I have a unique personal perspective on this because we have a rare kidney disease in my family. My dad's had a transplant, so is my uncle. We've been part of a Broad Institute, Wake Forest kind of rare kidney study for years. And about a little over a year ago, they discovered the gene underlying our family's condition. Ultra rare. We were one of 5 families published in the paper, but through that process, I found out I was a carrier, basically like it's a cascade screening kind of approach. But there's an alternate universe where you do newborn screening and someone like me finds out 30 years before they might actually need, or 20 years before they might actually need the kidney transplant, that they're a carrier. And in that particular case, there's a potential prevention strategy that evolves around diet. But it just has caused me to think about the whole thing differently, because when you go through it yourself, you can ask that question of, under what circumstances would I have wanted to know this? And obviously the fact that I'm a geneticist would change that compared to people make that decision differently if you don't know about genetics and can't go and read all the papers, right? You might choose to know only when there's something you can really do about it. But it's a really complicated set of questions, I would say, for us.
Yeah, no, and it's a great example. And, in the the very first few genomes we did, actually, the first one was Steve Quake, a bioengineer who'd sort of built his own sequencing technology. The next 4 we did in terms of interpretation was a family. There were 4 of the first 10 people to be sort of personally sequenced on the Illumina platform. John West was an executive in Illumina and had been part of Solexa, from where you are. Invented there, of course, and John actually moved back there recently. But yeah, his family were sequenced, and we built a series of tools, not just to think about the inheritance across that nuclear family, and some of those tools we still use to this day in thinking about trios and quartet sequencing for undiagnosed disease, but actually a kind of, I like the metaphor of the dial. Like, we took the medically important information from those genomes and allowed each of their family members to sort of turn dials around what they would want to know. And specifically, the database for that was set up in a way that was where there was classifications or dials, if you like, for the severity of the disease, but also for the possibility of intervention. Because classical genetics tells us about diseases for which we have no intervention, that people could decide they don't want to know. I think most people, if there's a clear preventive tactic and a potentially devastating disease, they're almost certainly going to want to know that. But even there, we shouldn't assume. And I think we can. I think part of it is that we have now the genomes available at a sort of cost that is, in range for a lot of people, or healthcare systems could decide that it's a good idea. And then the technology to allow people to choose what they want to know. And I think the ability and the people's sort of understanding of the fact it's their data, and that if they want to know it, they should be able to know it in the form and format that they want to have it.
What do you see as the key things that need to be proved out, or things about the system that might need to change to reach a tipping point where we actually say whole genome sequencing at a certain age is economically viable in a healthcare system like the US? Because you can sequence someone and you don't know before you sequence them if they're currently healthy, whether the impact's going to be in a dilated hypertrophic cardiomyopathy risk, or a Parkinson's risk, or a Huntington's variant. But at some point, the numbers check out where you say, actually, we should sequence everybody. But I wonder if it's about how we actually get that paid for with the way the the system is structured. Do you feel like the health economic case is there already, but the system isn't set up to pay for it, or is there still work to be done to prove out that actually, yeah, we should be sequencing everybody when they're 25 or whatever, and just do it?
Yeah, it's a good question. I have over the course of the last decade, and many others have too, tried on multiple occasions to get the health economists interested to do a kind of whole genome modeling, like a
Yeah.
cost effectiveness sort of analysis across the whole genome. We haven't ever fully achieved it, although there's obviously very talented economists out there and modelers, and little pieces of it have been nucleated. I feel, some people have done it around polygenic risk scores, others have done it around specific, cardiomyopathies. Obviously that's my world. I've seen lots of cost effectiveness, and I kind of feel that in a way we have 1,000 cost effectiveness analyses, and each one of them, and shows most of the cases they do show cost effectiveness for discovering disease early. But I feel like we sort of need the power of bringing that all together in one model to be able to really speak to payers of large healthcare systems. So my strong suspicion is that data would be very favorable, and certainly as we've approached that through different avenues, each of the individual avenues looks pretty favorable. So I mean, it's almost like the logical conclusion that it's going to be more favorable if you do this. I mean, you think about, you have a whole genome, you have a polygenic risk score for any validated condition, of which there are dozens now, and many that are really quite powerful at the level of Mendelian diseases. And then there's the Mendelian diseases, and as we all know, rare disease, individually rare, collectively very common. I mean, almost essentially as common as diabetes. So I think the case is there to be made. Obviously, it would always be optional. People, who worry about, too much information or the privacy element or other things, they don't need. But the question is, if you want to have it, should you have it, and can you have it? Those are the questions.
Yes.
And I do think that the healthcare cost effectiveness is very likely to be there. One thing that I realized early in doing a little bit of work of cost effectiveness is that it's somewhat of a paradox of the idea of cost effectiveness analysis, which is that there are many things that can be individually cost effective, but that doesn't mean any healthcare system can necessarily afford them all.
Right. Yes.
And so, because of the practicalities of actually, a given annual budget. And then there's other challenges. In the US, for example, is a big challenge with the fragmentation of the healthcare system, where people bounce around from individual payers over the course of their lifetime. So it's tough for an individual payer to say, well, I'm going to gain the benefit of this preventive strategy, which leads to very short-termist thinking, unfortunately. Now, in the UK, of course, and many other countries, there's much better, more integrated, and even nationwide healthcare systems that really could realize that benefit of preventive care across the entire country. And I think for many people, the question, to get back to the earlier one, is, well, why wouldn't you begin to offer that in the neonatal period as a better form of newborn screening? And as if you really talk about, I mean, if we really believe in at scale, $100, whether it's a clinical grade and includes the rest of it, that's another question. But newborn screening is in the tens of dollars, and apologies for the US currency for your listeners.
People can convert.
Yeah. But so it's not quite at that level, but it's getting pretty close, frankly. And I suspect for very many people, those kind of numbers, I mean, we know this from our clinics, that those kind of numbers are within range often in terms for testing people are willing to pay, even out of pocket, especially if you think of the potential of a lifetime benefit of potential preventive information.
Moving out of the world of genetics a little bit, you've got quite a bit going on in your research. A big area is around wearables and AI, and also how that fits in particular in going deep into cardiology. I noticed you have probably an Apple Watch or something that you're wearing. I'd love to hear a little bit of your perspective on what's the other frontiers that are really exciting to you right now across spaces of technology besides genomics.
Yeah, I mean, I think that we got into genomics obviously because of an interest in genetics, but what happened was that then it became kind of the tip of the spear, in some ways like the frontier for medicine of big data. Yeah. Because most of data was pretty small before. I often use this example as a cardiologist. Typical test is a lipid panel, which has, 5 numbers in it. So when we switch over to thinking about genomes, suddenly we need to think about compute and storage and 6 billion data points per individual. Is very different, but we're all nerds here, so we like data. and we're comfortable with that. So I think then when the aggregation of data, genomics together with the electronic medical record, for example, was the obvious beginning one, then we were often the people that were kind of tapped to be part of those conversations. And then as that data became the training data for new artificial intelligence approaches or digital tools like watches, wearables came into play, I think, again, we were the obvious group to ask because we'd set up compute infrastructure that could deal with large scale data. So I think that would be, I mean, when I think about the trajectory of us moving into that world, that's probably the why. But then the rationale is also, I think, fairly compelling if we think about thinking about precision medicine broadly, which, that the idea strictly, I guess, of that term is thinking about disease at the deeper level where we create subsets of disease categories because we understand it at usually a molecular level more deeply. But of course, it doesn't have to be a molecular level. And you can understand diseases using whatever data you have. The UK Biobank's been amazing for this, where people have really understood data that includes genetic data or now includes proteomic data, but can also just be defined on the basis of clusters of patients according to how they present. You can do precision medicine without a molecular tool or even a digital tool. But I think we've found that the digital tools in cardiology have been among the most useful and game-changing. And so whether it's Apple or Fitbit or Garmin or of course, there's lots of different companies now, the idea of monitoring your heart rate with photoplethysmography, which is just shining an LED light into the skin and looking at the reabsorbance coming back, allows you to determine in any moment what a heart rate is from a watch that somebody can wear all the time. And so when we think of a condition like atrial fibrillation, which is up to 10 or even 15% of the population as people get older and predisposes to stroke, which is among the most debilitating of conditions and accounts for the largest dollar amount of money spent on rehab in the US at least, and I think this would be the similar in other countries, then you get to the point of, well, why are we waiting till someone has a stroke to think about that?
Right.
And it's because the prior technology was something you put on for 24 hours. I mean, it seems laughable now, but we would literally put a 24-hour monitor on a patient and then say, come back in a year and we'll give you another 24-hour monitor. And maybe that's extended out now to 2 weeks or 4 weeks. But obviously a watch is capable every day and every night of monitoring your heart rate. And in this case, to pick up atrial fibrillation, you only need to tell it between a regular heart rate and an irregular heart rate. So it's not even a fancy AI algorithm required.
Right.
It's the standard deviation of the interval between your two heartbeats. And so what Apple, and we worked with Apple at Stanford, but so we know more about their approach and we collaborated there on that. But other companies have similar algorithms that can now pick up atrial fibrillation, meaning that you could get a preventive therapy, in this case, an anticoagulant, that could prevent your risk of stroke. So from a cardiovascular perspective, I think the digital tools can be really helpful. The other areas, musculoskeletal disease and neurological disease, especially neurodegenerative disease, I think digital tools also transformative, picking up things like Parkinson's disease and Alzheimer's disease earlier than otherwise. I think this just a really interesting area where technology really can make things better.
If there's another area of research that I'm really curious about, because going through your work and your website, there's domains that I'm kind of understanding how they connect together: medical genomics, cardiology, AI. But you also have this really interesting pillar of human performance and research studies focused on people kind of pushing the limits of human performance. I'm curious to hear the backstory behind that. How did you get interested? And maybe you can talk a little bit about what you're doing.
Yeah, of course. I mean, I have been always interested in sport and exercise, and growing up I played rugby and football and badminton and a few other sports. So it was always kind of sporty and fascinated by human physiology in many ways. That was part of what drove me towards cardiology. I just was fascinated by the heart.
Oh, interesting.
And how it responded to the stress of exercise. And so I've always been interested in that. And I think that then, and so some of my early work was actually in exercise physiology and exercise testing humans. And then most of the last decade or two, I've been focused more on genomics and molecular tools. But I always had this idea in mind that one day we could bring those tools to tell us more about understanding human performance. And there are these stories that I think, we talked about the story of little baby at the beginning, the kind of pivotal story in some ways for the world. And it's certainly the rationale for our study that I can mention briefly called ELITE, where we're sequencing literally the fittest people on the planet. was this cross-country skier called Eero Mäntyranta from Lapland, who won all these gold medals across multiple Olympics. But every time people measured the hemoglobin level in his blood, his hematocrit, they found to be very high. So he was under suspicion a lot for blood doping.
Right.
But in the end, he was found to have a genetic condition.
Just natural blood doping.
Right? Where he had a mutation in his APO receptor.
Fascinating.
And basically his body was just making a whole bunch of red cells. And so when that was found, he was sort of, this cloud was lifted from him.
Right.
But he always said, I don't do that. I don't do blood doping. But the only way to tell someone's blood doping is to say, do you have more red blood cells than you should have? So in a way, yeah. So this was sort of natural, like performance enhancement. And that just as a sort of story, a paradigmatic story, led us to the idea that, wow, we could learn a lot. If you think about what you learn about about driving cars,
yes,
from Formula One, that's obviously the absolute height of engineering. And none of us are driving Formula One cars, but we are driving cars that have insights that have been learned from studying and optimizing those cars. And so it's part of that study, Elite, where we're basically trying to find the fittest people on the planet based around VO2 max, so aerobic fitness in this case, rather than strength. And since cross-country skiers are often at the top of that, and Eero Mäntyranta was kind of the inspiration there. The study is open to anyone in the world, and they can go to our website. If you just Google Elite Study Stanford, whatever, if anyone thinks they might qualify.
What's the cutoff? What's the VO2 max cutoff?
Yeah, well, so we started very high, like at the point where even gold medal winners, some of them were not qualifying. The male number was 75 and the female number was 68. We've loosened the criteria down a little bit. Of course, now people have VO2 max estimated from their watch as well.
Yeah, I've got it on mine.
Yeah. So we've loosened that a little bit downwards, but also we're now accepting watch-based VO2 max estimates and also 10K times to try and just broaden the group. And we've been sequencing those individuals and we'll continue to do that. We have some pretty interesting early findings that we're just starting to kind of really analyze in detail right now. But it's been really a sort of fascinating thing, and watching some of the people who we know to be in the study compete, on international stages is quite something.
Yeah, that's fascinating. Is there anything that you published yet that you can share, or is it forthcoming in the near future?
Yeah, it's forthcoming. We're literally sort of finalizing some of the analyses right now, so we haven't too many things to share. But it's been an interesting world because there's been, this well, but where genetics was for lots of disease discovery at the beginning was just in a world where the numbers were too small and reproducibility was an issue.
Yeah.
And maybe not surprisingly, that's been a challenge in the sort of human performance arena. We've been able to do studies with a few hundred people and maybe small numbers of thousands, and that just doesn't really give you the power for replicability that you need. We, of course, have this combination of an extreme outlying kind of group of people, which means you can do more with fewer individuals. But we also now, of course, have UK Biobank and all of us and other population-based biobanks where we can then sort of map. We have that for control samples, but we can also sort of map findings in the extreme population to maybe more common variants in the larger population. So we're working on those kind of things. And then also we are part of a big NIH study called MOTORPAG, which is maybe the best acronym of any consortium I'm in, but molecular transducers of physical activity. So that's not about genetic discovery for physical activity, but rather about building a molecular map. So it's a multi-omic study of thousands of individuals as well as animals to try and understand the molecular multi-tissue changes associated with exercise.
One of the I'm going to just speculate wildly here, but one of the things that I find really interesting about the study, there's two things really. One is I'm wondering, one of the things we've learned about obesity and other cardiovascular phenotypes is there's a body and there's a brain phenotype, right? Like, and I wonder the same thing about elite performance, because you can definitely expect the examples that you had where their body is blood doping basically, and it does it naturally. But I also wonder if you also hear about these athletes who are the first in the gym at 5 a.m, they're the last to leave. And there is are they wired to just be so competitive and so driven? And I'm super interested to see what comes out of that. The second piece is just the fundamental biology that you learn when you zoom in on these extreme populations. There's often, something that is generalizable to just our knowledge of molecular biology more generally, right?
I think that's our hope. Yeah. This, of course, by studying the extremes of anything, you hope to learn about
Yeah.
gain insight that would be of benefit for a wide range of people. I mean, we of course use erythropoietin, not just because of that cross-referencing skier, but that was one of the studies that contributed to our understanding of the erythropoietin, which we use now in replacement therapy, of course. And yeah, I think that this idea of brain and body is I think fascinating. And there's different elements even to the brain, like you said. There's the people who are just really driven. Then there's another facet, which is, I mean, to what extent can you tolerate pain?
Yes. Yeah.
So I've been interested in that. And then a third one, which we've actually tried to do some analyses of, we have some supportive data, which is that everyone is aware to some extent of feeling a bit better after exercise, most people. And some people get that more extreme. And we talk about the exercise high, which I think people often talk about as sort of the opioid axis and endorphins. Actually, there's a nice study suggesting it might be more of a cannabinoid effect rather than an opioid effect, which is interesting sort of in and of itself. But regardless, I think that is a lot of the reason that people go back to physical activity and go back to exercise. And so I do think, as well as what the two areas we mentioned for performance, there's also maybe a kind of brain chemical predisposition to enjoying exercise. And every time I watch my dog like joyously run around the park, I'm thinking these animals are wired for the joy of exercise. And then there are humans, I think, wired that way too, which is only going to be to their ultimate, benefit from all the health benefits of exercise.
Yeah, that's right. The point about tolerating pain reminds me, I was a rowed competitively for a long time, and it's been years that I've been rowing before I got into a program that had a little bit more technology, and they did the lactic acid tests where you kind of row as hard as you can, and then they actually measure what is the lactic acid level in your blood. And it was amazing how the tolerance that some people had, they could get super high before they quit, whereas other people, like at half the levels or something, they tapped out and it was just too much. So that opened my eyes a little bit to feeling like maybe I just don't have the pain tolerance of some people to really get to the Olympic level, right? You got to be on another level.
Well, yeah, and I remember back when we lived in Oxford, around the time that Steve Redgrave was winning all his gold medals in rowing, there was a documentary, if you remember, about their 4, I think, and they were watching him on an ergometer in a gym, and he looked like he wasn't even working.
Yes.
Like, I mean, yeah, he was pulling obviously this huge amount of power, and then the minute he stopped, I don't know, he just bent over and writhed on the floor in pain.
Right.
And you realize that this is what he was feeling just 5 seconds ago.
Yeah.
He just was needing to win the race, which he, of course, won many times. And he was so controlled. But what he was feeling inside clearly was what you then saw, as he literally like curled into a ball on the floor, writhing in pain. So, yeah, it's always, that image is always stuck in my head and thinking about that sort of tolerance. And I think the pain of lactic acidosis is a very good example.
Yeah. More broadly, I want to ask the question about what you're interested in, but I want to ask it in a slightly different way, which is, suppose you had to drop everything you were doing and let's say you're restarting your lab from day one. Today, you've got your first R01 grant or whatever it is. Where do you go? What do you do?
Well, the most amazing thing about the life that we're lucky enough to lead in academia is that we actually get the chance to do that. Like you can do that anytime. Now, of course, you can't literally do anything because you usually need money, and so that means somebody needs to give you a grant. So it's not quite that, but I think that's one of the joys that we have in academia, that we have a chance at reinvention, every week if that's what you want. I mean, and so I would answer that by saying that one of the areas that we started working on in the lab is actually what I would start doing if I was starting today, but we're starting to work on it, which is this combination. And we've touched on very many elements of it over the course of our chat here today. But generative AI, right, is everywhere, mostly in the form of ChatGPT and these other models. But of course, that is a language model, and the language of life that we're most focused on are genomes and genes and proteins. And so our group has been more and more interested in the possibilities of generative models for helping us solve problems where the language we need is not English or Spanish or French or German, but it's DNA or protein. And so then I'm thinking, what are the problems to solve? I think we've come so far in diagnosing disease and understanding risk that the next decade, for me, and I think for many, is focused less on diagnostics. We need to get better at those and certainly need to make it more available. But I think we've come a long way. And so our focus is a bit more on therapeutics. How do we use that information to find like cures for diseases? And the medical profession has hated the word cures for the longest time because it was a ridiculous word when we're thinking of just palliating conditions that we don't really understand. But when we think about genetic disease, we really do understand it now. And with CRISPR tools and others, oligonucleotide sort of therapies, we actually have the ability to potentially affect what we might reasonably call a cure. And so that's, I think, very exciting. But despite decades of work, there are fundamental things to do with delivering those tools to the right tissue in the right place at the right time that we haven't solved yet. And so I do think thinking about what those tools are, optimizing those tools, and then optimizing the vectors for delivery are among the most important questions of our time. And I do think that is a place where generative AI can be very, very complementary. So we've started some new approaches in the lab where we bring those technologies. I obviously it's wet and dry lab, and this is a very nice combination. And I think when we combine that with the ability to have ready access to human tissue, which is something, again, in an academic medical center where we're doing heart transplants and liver transplants, and we can often get slice cultures of human tissue, that we have an interesting cycle then that we can go around where we can test those tissues, then teach the model, and test the tissues and teach the model. So I would say that that's something we're really excited about right now.
Yeah, that's fascinating because those kind of foundation models will be fundamentally constrained by the high quality data that you can put in, right? But if you have that feedback loop, then the sky's the limit.
It's the feedback loop that really counts. Yeah.
Yeah, fascinating. Well, this was a whirlwind tour. We covered a lot of ground. I just want to say thank you for taking the time with us, but also for all the amazing work that you do. It's incredible to see people like you doing all this impactful work across such a broad spectrum of things. So thank you.
Oh, you're so kind. And thanks for having me on. I really enjoyed the conversation.
My pleasure. And thanks everyone, as always, for listening. We'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.